Hi everyone, we're the team placebo detectors, and now it's our turn to present our mushroom edibility predictor. Our slogan is "Foraging has never been safer." Is also our goal for this project. Today we will cover these 7 parts in our presentation. So now I'm gonna pass it on to Lucas to start with the introduction.
Thank you very much. All right, so our product is a model that informs the user whether the North American gill mushroom is poisonous or edible based on the description of the mushroom's traits. It's established on the dataset from Kaggle. Its information of mushrooms is represented as letters like P for poisonous, D for descending, or G for grooves. And it's considered a classification problem. Before we changed the letters into integers for a better understanding, we decided to convert them into decimals between 0 and 1 instead. To solve the North American mushroom problem, we created 4 algorithms that can help determine the mushroom's type, such as the KNN, the SVM, the random forest model, and the neural network model. For data exploration, our features and targets were P and E, that was our target variable, for poisonous and edible. The original data set had 22 columns plus the target column, which is class. For cleaning, we removed 2 of the columns for accuracy and readability. And the size that we worked with ended up being 20 columns with 8,524 rows. Some more obscure mushroom features to know for anybody using the application can be seen here and referenced for anybody using the application. And I'll be passing it off to James now.
Thanks, Lucas. All right, so we created some visualizations to help us familiarize ourselves with the data before we started cutting things and changing things. And we picked the 7 of them that we felt would be the most helpful to just like understanding the data set and also understanding our process. So the first one that we have is a histogram that shows the proportion of poisonous to palatable mushrooms organized by cap color. Cap color is a good example of a variable that's not so important. It does an all right job of narrowing things down, but it's not something that the models prioritize because no matter which option is selected, it only does a moderate amount of work in narrowing it down to whether it's poisonous or edible. Next, we have the proportion of poisonous to palatable mushrooms organized by population type. And this is a good example of a variable that depending on which option is selected, for example, numerous or abundant, those are clear-cut. That will be an edible mushroom, essentially. But if something like scattered or several or solitary are selected, that's less sure. It could be either poisonous or edible, so it requires more work. Then we created a scatter matrix. I think this was one of the first visualizations we created. It was more of a jumping off point. As I understand it, the problem with it is just that it's a discrete set and there's only like at most 10 possibilities for each variable. And then there's thousands of data points that we have. So they overlap a lot and that can be kind of messy. Next, we have just a pie chart basically showing an example of that, how some options, when they're selected, really do a great job of narrowing down whether something is poisonous or edible. And then next, we have an example of brown cap color mushrooms. And this is an example of something that doesn't do a great job when it's selected of narrowing things down. Second to last, we created a histogram for odor, which is, I think, by far the most important variable for all of our models, just because so many of the options are like very like deterministic, I guess. Like it will be poisonous, it will be edible. And then lastly, we have a correlation matrix that we created with Plotly. The correlation matrix, it can be a little, I guess, misleading is the term, because if you scroll down, you can see that both odor and sport print color, they have like kind of whatever correlations with class, but those were actually by a pretty large margin the most important variables. And what our theory is that they both have a strong correlation with gill size, which is actually the variable that is strongest correlated with class. So there's indirect correlations that have a very large effect on whether something is edible or poisonous. That's all for this. I'll pass it off to Alan for data pre-processing.
Okay, so before we attempt to train machine learning models, we first have to clean up our data through pre-processing. We have to remove null values and irrelevant columns first. So within our data set, there are two columns which we have to remove. One being due to having too many null values, another due to containing only one unique value, which provides basically no useful data for whatsoever. So then after that, we have to label and feature encode our data, which turns our data, which is originally in which are all in strings originally, into numeric values, so that our machine learning models are able to understand it. And finally, we split our data into training test data, so that we can train our machine learning models and test them. So let's go look at our machine learning models now. So one of the machine learning models we use is KNN, or K nearest neighbors, and a description of the model is shown. So let's just look at the results. So we'll focus on the false negatives and the false positives because those are the errors for our machine learning model. So we see that our first test for KNN, which has hyperparameter of using 7 nearest neighbors, has around 11 sorry, we have no false positive errors, but we do have a few false negative errors, which are bad because a person who may use this model would see poisonous mushrooms as edible, which is something weird, so we reduce our false negatives. So we use grid search CV, and so it gave us a KNN model which uses a simplistic hyperparameter of only one nearest neighbor. But with that, we were able to perfectly predict the test data. So I'll give it off to Ozzy to explain the rest.
Thank you much. The second model that we use is a support vector machine, which is an algorithm to find the hyperplane in the n-dimensional space that distinctly classifies the data points. The model performs well with the hyperparameter of the C variable, which has a value of 5. That determines the size of the margin assigned by the model, affecting its accuracy and reliability. As you can see below, there's a feature of the image of a feature of importance graph highly shows that the model is prioritizing odor as the most important one to identify the mushroom. The third model that we use is the random forest. This works by creating multiple decision trees. Each is based on different proportions, runs, and select the answer with the most votes, shrinking the effects of mistakes. As you can see in the image below, you see the big random forest model. That is the final model that we made for the random forest. The hyperparameter is the n estimators, which determines the number of trees. It runs very well with no mistakes. There is another image with the feature of importance graph that also shows that the odor is being prioritized as the most important one to identify the mushroom. The last model that we use is a neural network, which is used to reflect on the human brain's behavior, allowing computer programs to recognize patterns and solve problems. Below it is a two-class PR curve, proving that the model is nearly perfect with the area of 1, which means the accuracy is really, really high. The model takes the input and processes them through layers, giving an output. And below is a similar curve, which is the ROC curve, proving that the model is also nearly perfect with the area of 1. As a result, the model works perfectly with the layer being 40 or more in one hidden layer. Now passing on to Marla for the rest of this presentation.
Thank you. So our interface allows the user to list the features of a mushroom. As you can see, our instructor is doing that. The output will be a table of the values you listed, along with a prediction on if the mushroom is poisonous or edible. And that will happen once you hit submit. As you can see, here's our table and our prediction saying that it is poisonous. In conclusion, all 4 algorithms performed really well, neural networks performed well as well, but it took a little longer. But with each algorithm, we found that odor, spore print color, gill size, ring type, and bruises are the most important features to identify a mushroom. And this is our team. And yeah, thank you for listening.
Yeah, that's all. Thanks for listening.